this is the Biology Paper 2 Higher from 2023 that I'm just now going to do the walkthrough for. Right, question number 1. Sexual reproduction in humans involves the production of eggs, egg cells and sperm cells. Name the type of cell division that produces egg cells and sperm cells. Okay, well, there's only two types of cell division. There's mitosis and meiosis. So M-E-I-O-S-I-S. Now it's one of those words that you actually have to spell right. And if you don't spell it right, then you actually don't get the marks for it. So be absolutely precise on that one. Sexual reproduction produces offspring that are genetically different from each other. Okay, so genetically different. Good. Give 2 reasons why sexual reproduction causes variation in the offspring. Okay, so what you've got is you've got the gametes, so that's your eggs and the sperms, are not identical. Okay, and you're getting genetic information from both parents. Okay, so your egg and your sperm, can't spell parents, your egg and your sperm, right, and then joining it together, right, and then what you're doing is you're going to get genes from mum, getting genes from dad. Okay. Polydactyly is an inherited disorder, right? The allele for polydactyly is dominant, right? Dominant. We've got to remember polydactyly is dominant. A person with 2 copies of the allele little d will not have polydactyly. A person with the genotype big D is homozygous, because homozygous means there's 2 of the same. What word describes the genotype big D little d? That is heterozygous. Okay, so this one, if you add little d, that would be homozygous recessive, and it's big D, that's homozygous dominant. 1.4. A person with genotype big D and a person with genotype little d plan to have a child. Determine the probability that the child will have polydactyly. Complete the Punnett square, so I've got to do that. Identify only offspring genotype that would have polydactyly. So I need to come back to that question a little bit. So what I'm going to do is I'm going to do the polydactyly at the top, and then, oh God, I'm doing it wrong, and then I've got little d. Then what you do is you cross over. Don't know why I changed the colours. So big D, little d. Then I've got big D, little d. So I'm doing there. Little d, little d. Now let me just go back to the question again. Complete the Punnett square diagram. Done that. Identify any offspring genotype that would have polydactyly. Okay, now I'm not quite sure what it means by identify. Okay, but what I'm going to do is I'm going to say polydactyl, and I'm going to say that one has and that one has. So that has and that has. Okay, so I need to identify any offspring genotype that would have polydactyly. Okay, they're the ones. Probability that a child will have polydactyly, 2 out of 4 is 50%. Okay, so we're always doing it as like a percentage. Okay, so embryos can be screened for the alleles that cause inherited disorders. So that's looking for then the genes that are then in there. So what you could have is you could have an embryo, right? And then what you could do is you could check out what genes are in there, what alleles are in there, to see if it is a possibility that they're going to have the disorder. Give 2 advantages of embryo screening. So an advantage could be, well, we'll talk about advantages in a minute, but if you think disadvantage, a disadvantage could be the fact that it costs a lot of money. And also it is kind of playing God. It's looking at the actual genes in the embryo before the baby is actually born. But 2 advantages. First thing is we could find out if the embryo has the disorder. Okay, so the first stage is we could actually find out if it has got it or not. The second one could be prepare for a baby with the disorder. Okay, so if you know that your baby is going to have an extra finger, then what you could do is you can kind of mentally prepare the fact that your baby's going to have an extra finger. If you've got a baby that's going to get cystic fibrosis or potentially get cystic fibrosis, then that's much more serious because that's a much more kind of long-term illness. So you need to be able to prepare and get mentally, I guess, ready for it. You also need to decide if you are having the baby. Okay, so whether that's an advantage or not, right? You have then, if you find out it's got it, then what you need to do, you then have to make a decision about whether you're going to keep it or not. But one advantage is, if you then end up not having the baby with the disorder, there will be fewer with the disorder long term. Okay, because what's happening is the genes in the gene pool for that particular disorder are then decreasing. Question number 2. It is estimated that 99.9% of all species that have ever existed are now extinct. Okay, why is the percentage of species that are extinct only an estimate? Okay, all individuals of one species have the same genes. Well, that's a load of rubbish. Extinction is always caused by humans when it isn't, because the dinosaurs weren't. Humans have not found evidence of every species. Okay, we don't know. There might be some tiny little mouse, right, from 1 billion years ago, right, that we're not aware of. So, you know, we can't say that that actually existed or not. So it is just an estimate. What evidence is used to study species that have become extinct? Right. So what we're doing is when we're looking at extinction, okay, and there's only really one piece of evidence, is fossils. Okay, there's no YouTube back then, and you know, all those different things. So it's fossils out of rocks, and that gives us an idea about what animals were around at the time. 2.3. A bacterium called Clostridioides difficile, C. difficile, that'll do, right, can infect the human digestive system. C. difficile can multiply and produce toxins, poisons. Okay, the toxins cause diarrhea. Nice. Doctors are concerned that new strains of C. difficile may evolve. Antibiotics may not be able to kill these new strains. Okay, antibiotic immunity. Okay, explain how the evolution of antibiotic-resistant C. difficile can be slowed down. Okay, so it's not asking what antibiotic resistance is, right? It's asking how can we actually slow down, because they're kind of saying, well, it's going to happen, right? How can we slow it down so it doesn't happen as quickly? All right, so we need fewer prescriptions of antibiotics, so less exposure. Okay, so I'll see this here. Explain. What word am I using? So. Okay, or I could be using because, but so works quite well. If you've got it, do not treat mild, mild infections with antibiotics, so the body can deal with it. Okay, that kind of makes sense really, doesn't it? Because then what you're doing is then it's not getting exposed to it. Don't use antibiotics. Okay, I'm just going to write AB on virus, as antibiotics don't affect viruses at all. Always complete a course of antibiotics so all bacteria are killed. Because if there's a couple left, right, then they've got a pretty good chance of actually being immune to it. Right, but the main part of this is it's an explain question. And what we're doing is we're saying so, I put as, so. Okay, so what it's doing, it's coming up with a fact, and then you're then saying so this is then happening. And we're talking about slowing it all down. Figure 1 shows a river next to a field. That sounds like a geography exam now. Okay, so there's a river, there's a field. Okay, pretty complicated diagram. Right, describe a method to investigate how the distance from the river affects the number of different plant species. You should explain how to use a transect in your method. Okay, I'd already kind of worked that one out, right? Investigate, distance from the river, number of different plant species. Okay, we already knew that was going to happen. Okay, so what we need to do is we need to measure the transect line at right angles to the river. Okay, then place a quadrat at intervals along the transect. Now I'm just going to flick up here because I'm kind of running out of space a little bit. Count the number of plants in each transect of quadrat. Okay, I would then always say record on a table. Okay, that kind of helps, and then repeat in 3 different places. And then what you could do, you can then do a mean. Okay, students use a valid method to investigate how the distance from the river affects the number of different plant species in the field. Right, so we've got a number of different plant species, we've got distance from the river in meters. Okay, mean and maximum and minimum. Okay, so we've kind of got a bit of an uncertainty going on here based on these box plots. Okay, what is shown by the data in figure 2? Okay, fewer different species are always recorded nearer to the river. No. The mean value students can be most certain about is 5 meters from the river. So that's where the lines are the shortest, so that is then correct. Cows walk on the ground near the river more than they walk on the ground further from the river. Okay, that kind of makes sense. Which is a abiotic factor that could affect the number of different plant species found near the river? Which is an abiotic. Abiotic is the non-living. Okay, microorganisms near the roots. No idea what that's talking about. Moisture levels in the soil. Yeah, that could affect it. That's abiotic, right? And it could affect the different plant species found near the river. Oxygen, unlikely. Primary consumers in the field, again, unlikely. Right. Question number 3.4. Increasing numbers of cows are being farmed across the world, right? We're talking about the world now. Okay, we're not talking about that riverbank. Explain, so let's be using lots of so's, the environmental implications of increasing the number of cows being farmed. Okay, so really what we've got kind of here is we've kind of got 2 questions, right? It's all to do with the numbers of cows and the environment. Okay, right, so let's start off and we'll break it up into what are the consequences of more cows. Okay, so what there'll be is there will be fewer plants. Okay, there will be trampling. Okay, because the cows are then going to trample all over the plants. There's less plants, so less, that means less. So less photosynthesis. Okay, more cows, therefore increase in carbon dioxide. Okay, then so they're the consequences of having more cows, right? Then we've also got the environmental implications, which is in this next bit. What will then be the environmental implications? Okay, so the environmental implications are if the carbon dioxide goes up, the greenhouse effect increases. If methane goes up, because that's what also cows produce, if methane increases, greenhouse effect increases. Therefore, if the greenhouse is increasing, global warming increases. And then it also means that because you've then got more cows in fields, there's less land for crops. Okay, question number 4. Sea lice, sounds yummy, are small animals that feed on the skin and the blood of salmon fish. Nice. Figure 3 shows sea lice attached to the skin of a salmon. Oh my god, they look grim. They look like little tiny octopuses or something. Of a salmon, Oncorhynchus. Okay, what is the genus name of salmon? Right, so if you think about right at the very bottom there, right, on the kind of kingdom one, right, so it's kingdom, phylum, class, order, family, genus, species. Okay, now the way that I remember that is King, oh, I'll do it in a different colour, let's make it exciting. King Philip, that's how you spell Philip, can order 5 giant sausages. Okay, so that's a way of remembering it really. Okay, what is the genus? So the genus is then the second to last one, right? That is then the one that's always done in italics. So what I'm going to do, I'm not even going to say it, onk, even though I started to say it then, Hynchus. Okay, it's obviously pronounced wrong. Which domain are sea life classified in? Okay, well, they're definitely not a plant, right? Not that makes much difference. They're definitely not a microorganism sorts. So what they are is they are eukaryote. Okay, they're a eukaryote. 4.3. Some salmon have genes that result in fewer sea lice being attached to the skin. Fabulous, that would be very useful. Describe how fish farmers can selectively breed salmon that sea lice cannot attach to. Right, so what we're going to do is we're going to select the fish that sea lice don't, what's the word, attach. Okay, that kind of makes sense, doesn't it? Breed them. Okay, so you get those fish that the sea lice don't attach to, and then breed the fish that they don't attach to. Then select the offspring that they don't attach to. God, blimey, what's going on here? Attach to. Breed them. Then repeat over generations. And eventually the sea lice numbers will decrease massively, and hopefully eventually what they'd do is they'd disappear completely. 4.4. Explain the advantages to salmon farmers of producing salmon that do not have sea lice attached to their skin. I can tell you what, if I knew that the sea lice were being all over salmon, right, then you would seriously think about whether you're going to have them or not. Okay, so no sea lice damaging the skin or feeding on the salmon. Okay, again, if you saw a salmon, right, what it had big chunks taken out of it, if that's what the sea lice do, you wouldn't be as happy about buying it. Also, the salmon, if they haven't got the sea lice on, will grow faster and larger, because all of a sudden the sea lice isn't taking away the nutrients that it's got. So salmon can then be sold for more. Okay, I always think about money, right? Because money, if it started to talk about salmon farmers, right, then what the farmer wants is money, all right? So it's always kind of a good thing just to think, if you're looking at, if you're seeing a sentence in a question, something that could be money orientated, like manufacturing or business, salmon farmers, postman, whatever it might be, right, then what you could do then is you can talk about money at some point. 4.5. Explain the disadvantage of selectively breeding salmon. Okay, right, so if we do start selectively breeding them, right, then all the fish are genetically similar. So they might all have the same inherited disorder. So they could then be prone to a new disease. Okay, so if you think about it, right, all of a sudden what you're doing is you're almost breeding loads and loads of salmon from very, very few numbers. What they're then going to do, they're going to keep the same genes, right? And those genes are then going to go all the way through, and you're getting rid of variation, because variation is also good. Question number 5. Okay. In vitro fertilization, IVF in glass, can be used to treat infertility. Fantastic. Which hormones are given to women having IVF treatment? Right, it's FSH, follicle stimulating hormone, and LH, which causes ovulation. Name the target organ of the hormones used in IVF. Right, so in IVF, right, the two organs that are then produced, right, this is a person, obviously, okay, those two, FSH and LH, are produced in the pituitary gland, which is right smack bang, it's kind of like a P-shaped thing. I'm just going to put a P there, pituitary gland right in the middle of somebody's head. But what they do is their target organ is the ovaries. Describe why microscopes are needed to process IVF. Okay, well, they are incredibly small. So you need a microscope to select the eggs. You need a microscope to select the sperm, because they're all too small to see. And then you might need a microscope to select the embryo in the end. Okay, so microscopes, you only need to write one of them. Describe why microscopes are needed. So you've got to be able to see the eggs, see the sperm, and then select the embryo. 5.4, right, there's a table here. Okay, so let's have a look at the table. Describe how the hormones given to women during IVF treatment interact with other hormones to prepare the body for pregnancy. Okay, right, so the hormones that we're talking about are FSH and LH, follicle stimulating hormone and luteinizing hormone. Okay, now what they do is they cause the production of progesterone or progesterone. They cause the production of progesterone and estrogen. Okay, and really what they do is they both are involved in maintaining the lining, so the embryo can embed and grow. Okay, 5.5. Calculate the simplest ratio of the number of women who have successful IVF to the number of women who have unsuccessful. So we're looking at successful against unsuccessful. Right, so if you look at the table, so the number of people that received IVF and the number of successful IVFs. Right, so what we've got here is we need to do a table. So we've got successful and we need unsuccessful. Successful IVF is 135. Therefore, unsuccessful IVF must be 450, take away 135, which makes 315. Then we need to do ratios. Okay, so I'm going to think about it now. What numbers go into both of those? 5 goes into both of them. So I'm going to knock that down to 5's going to 315, 63 times. 5's going to 135, 27 times. I can see that 9 goes into that now, so I'm going to do 7 and 3. So the ratio is 3 to 7. Question number 5.6. Suggest one factor that can affect the probability of a woman having a child as a result of IVF. So a factor that can affect the probability. So it's sort of looking at her chances. Right, so the older they are, the less chance it's going to work. The less fit they are, you'd assume that kind of more fit, then the more likely things are then going to happen. Obesity is also a factor that could affect it. The original reason. Okay, so if a woman can't produce eggs particularly very well, then that's obviously going to affect IVF. Okay, and also the number of embryos made. Okay, so if you do the IVF for treatment and you only get 2 embryos, then it's going to not be as successful as potential of getting 7. 5.7. Okay, give 2 arguments against the use of IVF. Do not refer to cost or religion. Okay, so other negatives could be the emotional and physical stress. Okay, because I imagine it is pretty tough going. A negative is success rate is low. And the other one is you could maybe get multiple births. Okay, so what you might do is where you might just get two embryos, make 2 eggs. Question number 6. Question number 6. The control of body temperature is an example of homeostasis. Okay, body temperature, homeostasis. All right. Give one other internal condition controlled by homeostasis. Do not refer to temperature. Well, it says other, so you're not going to do that anyway. 2 other main ones are blood glucose and water. 6.2. Explain. So I'm going to be putting some so's in here probably. Explain why the control of body temperature is important. Right? That goes back to the definition of homeostasis. The whole point in homeostasis is to maintain the optimal conditions for enzyme action. Okay, I could have put in there, so enzymes work more efficiently. I could have chucked in a so there somewhere. Where's the graph? I like graphs. The body temperature of a long-distance swimmers can change as the length of time swimming in cold water increases. Fair enough. Figure 4 shows how the body temperature of one swimmer changed in the first 4 hours of a long-distance swim. So they started at 37, and after 4 hours it's gone down to, looks like 35.5. Okay, let's have a look at the question. Calculate the mean rate of body temperature decrease per hour in the first 4 hours of the swim. Right, so started at 37, went down to 35.5. So what that is a 1.5. We need degrees per hour, so it's 1.5 divided by 4. Okay, so it's gone down by 1.5 degrees in 4 hours. So what we do is we make 0.375 degrees per hour, rate 0.375 degrees per hour. 6.4. Hypothermia is a dangerously low body temperature. Okay, for this swimmer, a 5.5% decrease in body temperature from the start of the swim will cause hypothermia, so 5.5%. Determine the body temperature at which this swimmer will start to have hypothermia. Give your answer to 2 significant figures. Okay, let's see where the 4 marks is coming from here. Right, so 37 degrees is kind of the body temperature that they're starting with. To find 5.5, I'm going to do multiply by 5.5 divided by 100 equals 2.035 degrees C. Okay, that's the amount 5.5% is of 37. So therefore, to work out the body temperature it goes to is 37 take away 2.035 equals 34.965. Okay, so that's what my answer is. However, we've got to then go back to two significant figures, and I look at two significant figures, and it's 34 is the two first significant figures. Look at the next number, which is a 9, so that then makes that 35. So body temperature to two significant, look at that, I've written it there again, is 35 degrees C. 6.5. Which gland secretes hormones to stimulate the adrenal glands? Okay, so a decrease in body temperature causes the adrenal gland and the thyroid gland to be stimulated, right? There can only be one gland that does that. Pituitary gland, okay, that's the master gland in the brain. 6.6. Explain the role of the adrenal gland in responding to a decrease in body temperature. So explain, so we're going to be doing lots of so's, right? The role of the adrenal gland, so we're talking about adrenaline, in responding to a decrease in body temperature, okay? How do we get our body temperature higher? Okay, so adrenal gland produces adrenaline. Okay, so that's kind of like a bit of a disclaimer. So we're talking about the adrenal gland, and we know that it produces adrenaline. Why does it do that? To increase heart rate. So more oxygen and glucose can get to active muscles, including the brain, so that more O2 plus glucose can be used in respiration to release energy. Right, so what we've done is we've talked about adrenal gland produces adrenaline, decrease in body temperature. Right, so adrenaline is produced to increase the body temperature by sending the heart rate higher, so that more oxygen and glucose can then get to active muscles to do more respiration, because then respiration releases energy. 6.7. Explain how the thyroid gland controls the response to a decrease in body temperature by negative feedback. Okay, so a decrease in body temperature causes the thyroid to release thyroxine. Okay, then what you've got to do is you've got to think, what does the thyroid gland do? The thyroid gland is all to do with metabolism. Okay, so body temperature goes down, thyroid then releases thyroxine. That then increases metabolic rate, I've got an extra letter in there, metabolic rate, which then causes heat to be produced by respiration. And then the negative feedback bit is, if the heat is then produced by respiration and the body then goes up, when back to the correct body temperature, there is less thyroxine, because the thyroxine then has done its job, and so what it's doing is it's feeding back into there, which is then the negative feedback.